All right, it's Monday. How are we all feeling? Yes, it's mid-semester Monday. I know, it's a rough one. So test is on Friday. And just as a reminder, Wednesday is a review day. Right? We will also have the scientific literacy assignment due on Wednesday. Come on in. Let's see, Wednesday, I also have that extra study session on Wednesday night that I will be here. I will have nothing prepared, but I will be here to answer all of your questions. But I believe Tri Beta and one of the tutors is also having a study session on tomorrow night. She sent out an email earlier, so I would check that out. So there's lots of study sessions for you to join. All right, let's finish up photosynthesis today. And this will be the last content for the exam, right? So as a reminder, we did, right, finish up the light reactions, right? So we went through this whole Z scheme. We saw how light was absorbed, how the electrons moved through different electron transport chains, right? And finally ended up in our molecule of NADPH. Right? Now in this process, we also generated that gradient of protons, which drove ATP synthase, so we generated a lot of ATP. Right? So we really have these three chemical products that have been generated so far. Right? And remember, the light reactions are only half the photosynthesis. I do have this nice little YouTube link on the corner. If you're a visual learner, this will walk you through the electrons moving through the molecules. We don't really have time in class to go through. It's like a 7-minute video. It does go into a little bit more detail, but I think it is a useful video to watch. So we have to start talking about the dark reactions now. All right, so we have produced NADPH. We have produced ATP. Oxygen has been a byproduct so far. Now in the dark reactions, this is where we are incorporating our molecules of carbon dioxide, of CO2, and making carbohydrates like glucose, right? And so we want to make these carbohydrates because they can be precursors to other molecules. So we can use glucose as a building block to build other molecules and other processes. But glucose can also be a great form of energy storage, right? You know, glucose can produce a lot of energy through cellular respiration. Right? So we can essentially store energy in our glucose molecules. But when we look at the dark reactions, right, and we're really talking about this cycle called the Kelvin cycle, right, this requires a massive amount of energy. Right? We need to use ATP and NADPH. Right? So we are using those products from the light reactions to drive the Kelvin cycle in our dark reactions. Now, ultimately, what will be produced is glyceraldehyde 3-phosphate. Does that molecule sound familiar? Yes, we also saw it in cellular respiration. So, glyceraldehyde 3-phosphate should be a familiar molecule for you. We saw it in glycolysis. So we'll be making glyceraldehyde 3-phosphate, which will then go on to produce molecules like glucose. All right, there's a lot that goes into the Kelvin cycle, but I promise you it's not too overwhelming. In fact, besides carbon dioxide, I'm only requiring you to know 4 molecules. All right? So this looks like a lot, but we'll break it down. The Kelvin cycle is really broken down into these 3 different phases, which is a different color on our Kelvin cycle here. The first phase is carbon fixation. And then we go on to reduction and carbohydrate production. And then finally, we're going to regenerate a molecule. We're going to regenerate RUBP. All right, so let's break these down. So phase 1, carbon fixation. Right? We are taking carbon dioxide and fixing it or incorporating it into the molecule RUBP using an enzyme called Rubisco. Right? So first step, we're taking our gas of CO2, incorporating it into our RUBP molecule, and what's produced what's produced is molecules of 3PG, all right, or 3-phosphoglycerate. Now in photosynthesis, I know 3PG, PG3, they sound really similar. I would encourage you to use the full name for those molecules because they can get easily confused. Happens in carbon fixation, and then it goes on into the reduction cycle. So this first step is one process, right? We're taking RUBP, we're adding CO2, using our enzyme Rubisco, which is not pictured on here, to produce 3PG. And that's carbon fixation, right? That is the first step. All right, does that answer your question? All right, so that's our first step, right? We have this one reaction. Our second phase is the reduction in carbohydrate production. And this is a series of several steps, right? But what I want you to know is that our molecule of 3PG is going to ultimately be turned into or reduced to G3P, or glyceraldehyde 3-phosphate. Right? So we're taking our 3PG, we're using energy, we're using NADPH. Right? NADPH is giving its electrons to these molecules, and we're producing G3P, glyceraldehyde 3-phosphate. Right? Now, one thing I want to point out here, as we're going through this cycle, notice we didn't start with one molecule of CO2. We started with 6, but each molecule gets incorporated into one molecule of RUBP. So we also need 6 RUBP. And this typically occurs in a series of 6 when you learn about photosynthesis. We're producing 12 3GP, so essentially splitting this molecule in half to create 2 smaller molecules. These smaller molecules are being transformed or reduced into G3P, 12 of them as well. Right? I mention these numbers because at the end of reduction and carbohydrate production here, only 2 of these 12 G3P will be used to make glucose. Right? So we're producing a lot, but we're only using 2 of them to make a glucose molecule, right? Or other sugars, most commonly glucose. All right? Which then brings us to our last step. Right? At this point, we need to start this cycle over again, but we can't do that unless we have our molecule of RUBP. Right? So we need to transform the remaining G3P molecules, 10 of them, back into 6 molecules of RUBP. Now this is a series of steps which we're not going to go into detail on, right? But we do use up some ATP, and in the end we get our RUBP molecule back, which will then go on to combine with CO2 and start this whole process again. Okay? So really the 4 molecules I am asking you to know, right? I am asking you to know RUBP, that first molecule, that's where CO2 gets combined, right? Asking you to know Rubisco, which is the enzyme that facilitates the combination of CO2 and RUBP. I'm asking you to know 3PG, right, or 3-phosphoglycerates. And I'm asking you to know glyceraldehyde 3-phosphate. Right? Just those 4 molecules besides carbon dioxide. And that's all there is to the dark reactions. Right? So I think the dark reactions are much less complicated to understand than the light reactions. Right? We have these 3 phases. Right? We're fixing carbon dioxide into a molecule. Right? That molecule will be eventually transformed into sugars, but also be used to regenerate our RUBP molecule. All right? But in the process, we're using a lot of energy. And that energy is coming from our light reactions. I really want to emphasize the importance of Rubisco here. All right, Rubisco is that enzyme, right, that combines molecule of CO2 to RUBP. Right. The full name here, which I am not asking you to know the full name, just know the abbreviated Rubisco. But the full name of ribulose 1,5-biphosphate carboxylase/oxygenase. Right? This enzyme is super important. It's thought to be the most abundant enzyme on Earth. Right? Every organism that photosynthesizes will need this enzyme. And as we start to look into different variations in photosynthesis, we will see why this enzyme can become very important. All right, but let's take a step back. All right, let's fill out the blanks on my image that I have up here. Right? And it's also a great time to ask me any clarifying questions that you might have. I will also keep passing back some assignments. Because that's needed in the first step, right? That's part of the reaction. All right, I realized I did not delete those answers off of your slides. So you got an extra little bonus there, which will be very good for studying anyways. All right, so I had you fill in pretty much everything that I might expect you to know for the exam, minus Rubisco. So Rubisco wasn't one of those fill in the blanks. All right, so notice how much simplified this looks compared to that initial Kelvin cycle diagram that we looked at. Any questions? Yes.
I thought for the first phase instead of
Yes, it does. Yeah. I have a mistake there, don't I? Yes. Just a little one. This should actually be down here, and this is 3PG, not GP, which is why I say use the full name for these molecules because they're so easily confused. All right. I will make a note here to go in and correct that on both your slides and mine. That way, when you're studying, you will be studying the correct thing. But thank you for that correction. Great observation. All right. It's 3PG. Right? I know, they're easily confused. Any other questions? All right. Well, let's move on to learn some of these variations in photosynthesis. Right? The environment is unpredictable. Right? What works for one plant is not going to work for the other plant. Right? So if we look at different environmental conditions that can change, we can change in light intensity, right? So think about how little light we receive here in our northern part of the hemisphere versus at the equator. Right? Temperature can change, think seasonally, think also where you are on the globe. Also water availability, right? So we kind of had a dry summer, right? But think about plants that live in the desert that always experience drought, right? So these conditions can influence a lot of how the plant works, and we're really going to look at how it influences photosynthesis. So plants have to be able to balance water preservation and CO2 delivery. So if we think about back to our leaf anatomy, right, we talked about the stomata. Right? These stomatas, they are pores on the leaf surfaces that allow gases in and out. Okay? Now that's important for photosynthesis because we need to allow CO2 to come into our cells because we incorporate that in our Calvin cycle. Okay? But when these cells are open, we also allow water to exit. Right? Water exits as well as oxygen, that byproduct of the light reactions. Right? Now this process of water being able to exit through the stomata it's a pretty important process in plants. Right? So we want to allow water to exit. But if these plants are in an environment where it's really hot or really dry, this water, when the stomata is open, is going to leave at an excessive rate. It's going to leave too fast. And eventually, the plant could die from dehydration because it's lost all of its water through the stomata. Right? So especially these plants that live in desert-like conditions or conditions that are just really dry, right? They leave their stomata open, they're going to dehydrate. So we have to have a way for plants to be able to keep their stomata open to receive carbon dioxide for photosynthesis, right? Without letting too much water out. Right? So when we close the stomata, right, we're not going to let water out, but the CO2 delivery for the Calvin cycle, for the dark reactions, are going to stop. However, right, the light reactions, they don't rely on CO2, right? They only rely on the sunlight. Sunlight doesn't need to enter into a stomata, so the light reactions can still continue. The light reactions are continuing, we're continuously producing oxygen. Under light reactions, they get that electron from water, they split water apart and create oxygen. So we're increasing oxygen levels and decreasing CO2 levels. And this can lead to something called photorespiration. All right? I'm going to explain photorespiration in a couple more slides that need just a bit more background information before it'll make sense. Yes? Most of the time? Great question. I don't know the specific mechanism. I would assume that the plants there are just adapted to these low light levels and are able to produce enough. I don't know. That's a very interesting question, though. I don't know what it is. I'm just assuming it has something to do with adapting to lower light levels, or maybe they just have kind of like winter here, right? They just go dormant for those months. That would be my guess. All right, so to understand that photorespiration, we need to understand that enzyme rubisco even further. Right, so normally, and what we have talked about up until this point, rubisco is the enzyme that allows CO2 to combine with RUBP to make our product of 3PG. Right? That's what normally happens. That's what we've seen in the Calvin cycle thus far. Right? Because the molecule 3PG has 3 carbons, right, we call the plants that undergo this photosynthesis a C3 plant. Right? Now C3 plant, most common type of photosynthesis we have learned so far, most plants on Earth are C3 plants. Right? All the trees that you see outside are going to be C3 plants. The problem is the enzyme Rubisco, these active sites can also bind to oxygen. Right? So normally it binds to CO2, and everything is all fine and dandy, everything works like it should. Right? But if the stomata are closed and there's no more CO2 coming in, but we're producing a lot of oxygen, now that oxygen is actually going to bind to the active sites on Rubisco instead. Right? And so when this happens, things become really inefficient. Right? So if we combine RUBP with oxygen instead of carbon dioxide, we're still producing 3PG, but we're only producing one molecule of 3PG. This can be used in the Calvin cycle as normal. Right? But we're also producing this other molecule, 2-phosphoglycolate. Right? And eventually, that molecule has to be processed. It'll release CO2, and it uses ATP in that processing. Right? The big picture here, this is really inefficient compared to when we use CO2. Right? Now, Rubisco binds better to CO2, right? So it's normally going to bind to CO2. If there's oxygen and CO2 present, it will prefer to bind to that CO2. Right? But since if we have to close that stomata and CO2 decreases and oxygen is now high, it will bind to oxygen instead. And this is what causes photorespiration, right? This equation here is what we call photorespiration. Right? So super inefficient. So plants that might have to close their stomata or risk being dehydrated have to deal with photorespiration. They have to find ways around this. And this is where these different forms of photosynthesis come into play. Right? And I know this is the last of the content before the exam, so really focus on big picture here. So we have plants that can undergo C4 photosynthesis, which spatially separates out these processes, or they can undergo CAM photosynthesis, which temporally separates out the processes. Right. So let's look at C4 first. The big thing with C4 is it's spatially separating out where things happen. So if you remember our leaf anatomy, right, we have those cells in the middle that we call the mesophyll cells. And this is where photosynthesis typically occurs. Right? But in a C4 plant, we have the addition of a specialized cell called a bundle sheath cell. See cells in yellow up here. They typically surround veins in our leaves. Right? These bundle sheath cells, right, is where the Calvin cycle occurs. So if we look at C4, and I'm going to turn to the next slide because I like this image better. It's much more simple. If we look at the C4 pathway, we have carbon dioxide coming into our mesophyll cells. But instead of Rubisco, now we're using an enzyme called PEP carboxylase, which combines CO2 to the enzyme PEP. This makes oxalacetate. Right? This should also be a familiar molecule for you, right? Yeah, used in the citric acid cycle. So this makes oxalacetate, right, which then will be transformed into this molecule malate. And this is all happening in our mesophyll cells. Our malate then moves into the bundle sheath cells and brings that carbon dioxide with it. So essentially, we're fixing carbon dioxide to a molecule that's kind of like a messenger molecule. It's hanging on to the carbon dioxide, bringing it to a different cell, and then it's giving that carbon dioxide away. Right? And by doing this, it concentrates the CO2 where the Calvin cycle is actually occurring. That way there's always going to be more CO2 than oxygen. Right? That reduces that photorespiration. The Calvin cycle will continue as normal. Right? We'll use Rubisco here. And what's left is pyruvate, which will be transformed back into this PEP molecule, and the cycle continues. Right? So we are spatially separating out where things happen. Right? And this spatial separation concentrates CO2 into the cell where the Calvin cycle actually occurs. All right, On the flip side of this, plants can also go through CAM photosynthesis, right? Also known as crustacean acid metabolism. I always struggle over that name. It was named after the family of plants that this was first discovered in, right? So CAM plants, right, will, instead of keeping their stomata fully closed or fully open, right, they're going to separate when they open up their stomata, and they're going to open their stomata at night, only at night, right? It's cooler at night, right? They're not going to lose as much water at night, so they can leave their stomata open at night. So in the nighttime, they can allow CO2 to enter. That CO2 will be fixed into a molecule called malate, which will then be stored in a vacuole as malic acid, right? So again, we're kind of using this messenger molecule, right? It's holding on to its CO2, storing the CO2 in the vacuole, right? Until the daytime when the sun comes out and we can do the light reactions, right? When that happens, our CO2 is moved out of the vacuole, given to the chloroplast, and can go through the Calvin cycle as well. Right? So stomata are closed during the day, but that's okay. We won't have as much of a buildup of oxygen. Well, we will have a buildup of oxygen, but it won't matter as much because we also have plenty of CO2 stored away for us to use. Right? So, big picture here, we're separating when things happen. Right? Daytime versus nighttime. All right, so these are both adaptations that help plants live in these really dry and arid environments. Right? They help them consume less water, and they help reduce photorespiration, which is a really, really inefficient form of photosynthesis. All right, but again, big picture here, we're spatially separating things out and versus temporally separating things out. Right? C4 versus CAM. And as a reminder, most plants on Earth, they don't do either. They are C3 plants. They do regular photosynthesis, like we've talked about in depth. Are there any questions? All right. I am going to say these are great review questions. And if you noticed on my last exam, some of the review questions that we did in class were ever so slightly tweaked to become exam questions. I'm going to encourage you to work through these on your own, but I'm going to skip to the content check so that everybody has enough time. The exam is coming up, and just some study tips. Review these class activities. They could show up on an exam. Draw out these processes. Right? Utilize the whiteboards around campus or the chalkboards in an empty classroom during the evening. Try to ask yourself potential exam questions. Right? Rewrite your notes. That's also a good way to study, and put it into your own words. Right? If you can put it into your own words, that means you understand the content enough to do so. If you have somebody you'd like to study with a partner. Even if they're from a different class, they should be studying the same material. And visit the tutors, all right? They are there even if you just need to say, hey, can you study off the study guide? Right? Which the study guide was posted last Friday if you missed my email. Right? On Wednesday, we will have a study session in class, right, that I'll have some things prepared for. And then that extra study session in the afternoon, as well as the Tri-Beta one tomorrow. But your scientific literacy assignment is due on Wednesday as well. All right, let's move on to the content check. All right. I wanted to make this one a little bit easier because I've given you so much information, and there was a lot that you could have drawn out. But instead, I'm simply going to ask you, where are these 4 things happening in a C3 plant? All right, so normal photosynthesis. I want you to be as specific as you possibly can be. All right, so yes, all of these things happen in a chloroplast. They all happen in a mesophyll cell. But can you be more specific than that?